Alright, we have come to the last video of the chapter on enzymes. Okay, good job. So this last video is actually on this concept that we call denaturation. Okay, denaturation. Now what exactly does it mean? Okay, first of all, I want to explain this term to you by drawing a link to what you are very used to already. Now, do you know what is this? Okay, I hope you know what is this cartoon. Okay, this is your Gudetama, right? Okay, but do you know that your Gudetama is actually just an egg yolk, right? And you see the egg white around it. Now, why am I talking about this? Because today I'm talking to you about an egg. Now, I believe all of you will have cracked or seen a cracked egg before at home. You will notice that there's an egg yolk that's yellow in color, and there's something that we call a certain term that's surrounding the egg yolk. That's what we call the egg white. Right, so it's very interesting because the egg white, when you crack out the egg, is actually not white in color. It's actually colorless, right? It has no color, but people call it the egg white. And some of you might argue that if I were to go and cook it and pan fry it, I get this sunny side up, and now it is really white in color. So the egg white is really white in color. That's why we call it egg white. Now, what am I trying to draw over here? Now, you notice that in your egg white, there's a lot of protein. The protein initially, before you heat it up at the frying pan, it was colorless. After you heat it up, it became white in color. So definitely a chemical reaction happened to the protein, right? And this chemical reaction is what we call denaturation. So we say that the protein in the egg white denatures under the high temperature. Likewise, if you think about this for a second, let's say I give you a piece of steak that is not cooked yet, and as you go barbecue it or sear it or pan fry it. After everything, you realize that the texture change, the color change, the taste change, the smell change, right? And this entire process, likewise, is because the protein in the meat, right? Meat has a lot of protein, actually went through this process called denaturation under the high temperature. Alright, so I've been saying this over and over again. Proteins, if you do something to them, like heating them up, can go through this process called denaturation. So, since this chapter is on enzymes, and enzymes are proteins, it means that enzymes can also go through denaturation, right? So the term denaturation refers to a change in the 3D shape, and this results in the loss of the active site of the enzyme or the protein. Now, what does this mean? An enzyme that's denatured will have lost its active site, and therefore this enzyme can no longer work, okay, because the active site shape will change. Now, what can cause denaturation? In your syllabus, there's two main things. Number one, high temperature. Extremely high temperature will result in denaturation. Number two, when you add in acids or alkalines and resulting in a change in the pH, and that can also result in denaturation. So, in a nutshell, imagine that your protein is like a ball of wires coiled around it. What denaturation do is that it actually unwinds the entire coil of wire to give you back your normal wire, which is straight, and then the shape is gone, therefore the protein can no longer work. So, what better way to imagine this than to just think of it as like a bench at the park. Now, imagine today you are walking along the bench at the park and you meet the guy or the girl of your dream, right? So you see that person and you really fell in love at first sight. Okay? But there's no way you can talk to this person. However, because both of you happen to walk past the bench, both of you sat down, and then you all became a couple. So imagine this thing happening, right? Okay, two of you walking along the park, you saw a bench, you all sat down at the bench, then you all started to talk, got to know each other better, and you all became a couple. Now, do you know that in this entire story, actually I'm trying to bring across the idea of your enzyme substrate and enzyme substrate complex? Okay, do you know what exactly is each of these things trying to represent? Okay, so for instance, what do you think the bench is representing? If you think about it for a second, the bench was what brought the two of you together, right? Allowing your two to sit over there. It has a bench that matches your buttock, right? It has a shape that matches your buttock. And this is therefore the enzyme. Now, what about each of you? What do you represent in this entire example? Yes, both of you are like the substrate A and substrate B, right? Coming by together. And when both of you are on the bench, you are actually like an enzyme substrate complex. When you start to get to talk to each other, get to know each other better, then eventually you'll leave as a product, right? So that's when you have the fairy tale, and that's it. So this is the idea of how an enzyme works. But if you want to think about denaturation, now try to imagine this. Imagine today you are still going to the park, but there was a fire. With a fire, the bench will start to catch on fire and the bench will be destroyed. So there is no longer a space for your butt to sit on, right? So the active site is gone. Or if you think about it, someone take an acid or an alkaline and they spill it over the bench, right? This causes a change in the shape of the bench, and therefore once again it affects you and you can no longer meet with the guy or the girl of your dream. So in terms of the diagram itself, right, this is a very good illustration of what is denaturation. Initially you have this enzyme in this shape and you have this substrate in this shape. So originally the substrate fits very well because the shape is complementary to the shape of the active site. Now when there's high heat or change in the pH, there's a loss or change in the shape of the active site because the enzyme goes through denaturation. Right, so now instead of looking like the original case, the enzyme actually looks like this now. One important thing that you realize is that number one, the shape changes totally. Number two, the active site is gone because now it becomes like this. If the substrate, the original substrate that was there, now tries to fit in, it can no longer fit in because the shape is no longer the same, or rather it's no longer complementary. So the substrate can no longer fit into the active site because its shape is no longer complementary to the active site. So it can no longer fit in, and therefore there's no enzyme substrate complex and there's no reaction that will happen. So this is what happens. These are consequences of denaturation. That's why it's important for us as bio students and teachers to understand what can be done to the enzyme. Now, there's always this statement that students always say, enzymes are killed by heat law or acid and alkaline. Now, do you think that this statement is true or false, that enzymes are actually killed by acid or heat and alkaline? Because I told you, right, heat and acid or alkaline can do something to the enzymes. But the important thing to take note is that although the original enzyme works like this, and in the presence of heat and pH, the enzyme can no longer function, this is not a right statement because we say that the enzymes are denatured by heat or acid and alkaline because denaturation is not the same as death. In fact, enzymes are not even alive. If something is not alive, it's just a protein. You know, the protein that is inside your meat, those are not alive, right? It's just a chemical. If enzymes are not alive, how can they die, right? So we say that enzymes are denatured by the heat or acid and alkaline. Now, I mentioned to you just now that there's two things that can cause denaturation. The first of it is heat. So let's go on a bit further to talk about how does temperature affects the enzyme itself. So over here we actually have a graph to show you how does the temperature affects the rate of reaction by an enzyme. So if you look at the graph itself, right, on the x-axis is the temperature, on the y-axis is the rate of reaction, which is the rate of enzyme activity. So basically, if over here on the y-axis is high, it means that the enzyme is doing a lot. But when it says 0, it means that okay, there's nothing that the enzyme is doing. Okay, so you notice that at low temperature, which is around this region of A, when the graph is still very over here at low temperature, whereas the graph is not very high up, right? So the rate of reaction is actually very low. This is actually because the enzymes are inactive. Now, what do you mean by inactive? Enzymes are inactive because they don't have a lot of energy at low temperature. So it's like very cold and the enzymes cannot really move. As a result, the rate at which they meet the substrate, the rate of collision with the substrate, becomes very low. And therefore, you don't really form a lot of your enzyme substrate complex, and therefore, the rate of reaction will be slow, right? So imagine a cold temperature, your enzymes are not really moving that much, they don't really meet the substrate, and then no complex is formed, and therefore no product, and the reaction will be slow. Now, as the temperature continues to increase, you realize that the rate starts to increase as well. This is because as the temperature increases, the amount of kinetic energy actually starts to increase. Now, what do I mean by that? The enzyme will start to have more energy to move around. Remember your solid liquid gas? Right, when you heat up something, they start to have more energy and they start to move around more violently. Right? So, same thing over here. Then the rate of effective collision between the enzyme and substrate, between the substrate and enzyme, start to increase. Right? And as a result of that, you start to have more and more of your enzyme substrate complex, and the rate of reaction will start to increase as well. In fact, there's this theory to say that for every 10 degrees Celsius increase in the temperature, the enzyme activity actually increased by about 2 times. Right? So instead of the original part whereby it's low temperature, from here onwards, you realize that as the temperature increases, the rate of enzyme activity starts to increase. It doubles at every 10 degrees Celsius because it has more kinetic energy now and there's more collision between the enzyme and the substrate. Now, you might realize that this graph suddenly shoots down, right? But before everything, there's this point whereby it's at the highest rate of reaction, and this takes place at this temperature, which we call the optimum temperature. Now, the optimum temperature, optimum means the best, okay? Best doesn't mean the highest or the lowest. The optimum temperature for different enzyme can be different. So, for instance, in your normal human, for the optimum temperature, about 37 degrees Celsius. But for an enzyme that's in an animal that survives in the desert or that survives in the volcano, then that particular enzyme, the optimum temperature will be much higher because the surrounding will only allow this kind of enzyme to be present. For most of the enzyme, the optimum temperature should be about 40 degrees Celsius to about 45 degrees Celsius. Alright, but like what I mentioned just now, different enzyme will still be different. Will still have different optimum temperature. Now, after the optimum temperature, that's when it gets interesting because suddenly everything goes downhill, right? It's like at the start, so nice when temperature increase, but after this optimum temperature, the enzyme activity actually starts to drop. Now, what happens when it's too hot over the optimum temperature? That's when you have your denaturation. The high temperature actually causes the bonds within the enzyme to break, and then there's a loss or change of the active site. So we say that the enzyme is now denatured and the substrate can no longer fit in. Okay, so the enzyme is now denatured and the substrate can no longer fit in, and therefore there is no reaction. Okay, so this is how you describe the graph of an enzyme, the temperature graph. At the start, when the temperature is low, we say that the enzymes are inactive, right? So when they are inactive, they are moving very, very slowly and the reaction is very, very slow. However, as temperature starts to increase, and kinetic energy actually starts to increase, and the rate of collision starts to increase, and everything goes well. When it reaches the optimum temperature, that's when everything is at the best state, when you have the highest rate of reaction. However, once you go beyond that, what you realize is that the enzyme starts to denature and change in the active site, so the substrate can no longer fit in, and there will be no more reaction, and this entire thing goes down. Okay. The unfortunate thing is that once the temperature increases beyond the optimum temperature, the enzyme is denatured, and that's it. It's the end game for the enzyme. There's no such thing as, okay, restart, enzyme go back to normal. The enzyme will be denatured and is useless from then onwards. Okay, so just to sum up, okay, beyond the optimum temperature, enzyme activity decreases because initially it was supposed to be like this, but now the enzyme looks like this. The active site is gone. So it's denatured and the substrate can no longer fit in, and therefore there is no reaction. Alright, the last one, which is on pH, to show how does it affect the rate of enzyme catalyzed reaction. Okay, so first of all, you need to know that acids refer to substances that has pH below 7, whereas alkalines are substances with pH above 7. Okay, so let's take a look at the graph in the case of an enzyme when it changes with a pH, right? So in this graph now, something that's different is that over here we actually have the pH scale on the base of the graph, which is the x-axis, and on the y-axis is still on the rate of reaction. But likewise, in this graph, we saw a highest point, right? This is what we call the optimum pH. So for this particular enzyme, the optimum pH is 7. Okay, so at the optimum pH, the enzyme activity is the highest. But do take note that different enzymes can have different optimum pH. So, for instance, because your stomach is acidic, the optimum pH of the enzyme there will be more acidic than normal. Okay, so it really depends on the surrounding and where the enzyme is found in. Now, the interesting thing comes when you start to move away from this optimum pH. That's when everything goes downhill. Right, so once you change the pH, it will result in a loss of the active site and a loss of the function of the enzyme. So we say that the enzyme is now denatured and the substrate can no longer fit in, and therefore the reaction starts to drop. Right, this is very similar to in the case of a temperature. Okay, so just a recap, at the optimum pH, that's when the enzyme activity is the highest, but once you change and move away from it, then the enzyme gets denatured and the substrate can no longer fit in and the reaction starts to drop. Right, so to sum up, initially it's supposed to be like this, the substrate fits in well because it's complementary, but once the pH changes, the enzyme actually goes through denaturation and the active site is gone and the substrate can no longer fit in. So this will help to sum up on the entire chapter on enzyme, whereby we actually talk about a few important things. So the definition of an enzyme, how they speed up chemical reaction without itself being chemically changed, how they are actually proteins, right, by nature. Then we move on to talk about the two types of reaction that an enzyme can catalyze, right? One of it is when they take smaller molecules, piece them together. That's what we call the anabolic reaction. Another one is when they take a bigger molecule and then break them apart into smaller ones, and that's what we call catabolic reaction. We then go on to talk a bit more about how do enzymes actually function. That's when we share about the three types of characteristics of an enzyme. Right? So enzymes helps to speed up chemical reaction, is required in small amounts, and then is highly specific in action. Then we start to talk about this concept called denaturation. Now, denaturation is something that we see in our daily life, from the frying of eggs to the cooking of meat, so on and so forth. When it comes to the topic of an enzyme, denaturation is best explained with this particular diagram that you see over here. How an original enzyme has a specific shape called an active site that allows the substrate to fit in. But once it goes through denaturation, this shape is lost and therefore the substrate can no longer fit in and there's no more reaction. And that's it for the enzyme. We went a bit further to talk about how do temperature affect the enzyme and how does pH affect the enzyme, and that's when you were introduced to the two different types of graphs. Okay, so the main thing that you need to understand for this chapter is that you really do have to practice writing, write out the questions, and try to put your thoughts into words. From then on, the teachers can actually help you to fine-tune your answer and look out for what are the keywords they are missing. And with that, we have come to the end of this entire chapter. See you back in class. Bye bye.